So it's my great pleasure to introduce our session moderator, Farida Sohrabji, who is the Joseph Shelton Professor of Neuroscience at Texas A&M University for this next session on brain development and evolution. Thank you.
Thank you for that. I'd like to welcome everybody to this next session, which is going to focus on brain development and the evolution of the brain, and specifically in the context of neurological diseases, both pediatric as well as adult. Our speakers today are Dr. Jenny Hsieh, who is a professor and the Seams Foundation Chair in Cell Biology at the University of Texas, San Antonio. The next speaker is Genevieve Konopka, who's an associate professor at UT Southwestern Medical Foundation, and then followed by Roy Silito, who is the associate professor at the Baylor College of Medicine. So we're going to start out first with Jenny Hsieh, and as an introduction to her work, Jenny directs a neural stem cell biology laboratory, and her research program strives to decipher the gene regulatory mechanisms that control neural stem cell fate and to apply these mechanisms to understand the causes of epilepsy and to develop better therapies. Please welcome Jenny Hsieh.
The pointer. Okay, thank you very much to Joe Takahashi and the organizing committee. I'm very delighted to be here to talk to you about some of our work. And also, I want to thank all of you for your interest. So I'm from UT San Antonio, and what I really want to talk about today is some new tools and technologies. This was actually raised as a question in the first session about how you might apply stem cell models to understand brain development and brain disorders. Okay, so the example I'm going to give today is epilepsy, and that's a childhood and also an adult disorder for the brain. And this is one example that I'm going to talk about today, but it could be a numerous number of brain diseases, as you can imagine. So epilepsy is very common, and so it's characterized by these spontaneous seizures in the brain. And in fact, it's been estimated that 1 in 26 people have active epilepsy in the United States. So it's very common, but and in fact, 1/3 of epilepsies are still refractory to treatment. So, and many of these are children. Oops, okay. So in general, seizures can come from different parts of the brain. For example, it could come from one part where you have seizures called focal epilepsy, but it could also come from different parts of the brain, and we call this generalized epilepsy. And some of the causes include traumatic brain injury, but today I'm going to focus on genetic causes of epilepsy. So the medicines that treat seizures, there's many, and most of them are, some of them are actually on this slide. So the drugs tend to work by directly on the ion channels expressed in the neurons themselves. So for example, if you have too much excitation, these medicines will try to reduce that excitation, or might try to increase this inhibition to try to bring the brain back to the normal state. So really, the first compound was discovered in the 1800s, and most of the drugs since then, first, second, third generation, have kind of built on this theory. However, still, there's about 400,000 patients that don't respond to any of these drugs. So this has led scientists to try to figure out, what can we be doing better? So really, we still don't really understand what are the complex, parts of the brain that really lead to epilepsy. And so since the advent of next generation sequencing, we've heard about that in the early sessions, people have begun sequencing genomes to try to identify if there's genetic causes. So here on this slide are just some of the genes. There's been about a couple, 1 to 200 genes so far that I've identified in patients with epilepsy. So today I'm going to really talk to you about one gene. It's this gene called ARX, because our first patient we recruited from UT Southwestern before, where I was at for the last 13 years, has mutations in ARX. So this is, before I forget to mention, I really want to acknowledge our team, the team of stem cell biologists and clinicians, we call them Team Organoid, to tackle this problem. So these are postdocs, I'm sorry, keep doing that. So postdocs, Vanessa and Parul and Zane in the lab, and we've teamed up with clinicians, Drew Thodeson from pediatrics at UT Southwestern, and Jay Schneider, internal medicine, and also together with stem cell biologists, Chris Navarra and Sean. Okay, so again, I mentioned this, our first patient is Hsieh. He has one of these genetic mutations in...
Okay, come here.
So he's 11 years old, he goes to public school, but he has seizures and he also has movement disorder and cerebral palsy. And Hsieh, unfortunately, does not respond to any of those medicines on my previous list. So really, once he's failed 4 or more medicines, the epilepsy doctors have determined that he's refractory, and he gets sent home, and there's really nothing available for Hsieh and other children like Hsieh. So what do we do? So what we decided to do is try something new, and so basically we recruited Hsieh into our study, and we made stem cells, and I'll tell you about that in the next slide. But really, from the genomic analysis of Hsieh, he has a mutation in this gene. It's a transcription factor. So there's, as all transcription factors, there's a part of the factor that binds DNA, but there's also a part of this protein that pretty much people don't really know what it does. So it's these blue domains highlighted here, polyalanine tracks. So they're repeats of alanines, these amino acids, and in Hsieh, it's repeated more than the normal number of times. And in other patients like Hsieh, these asterisks that are shown on these slides, it actually is linked to a host of different syndromes, for example, epilepsy. Okay, so in Hsieh's brain, if you did an MRI, you actually won't find anything structurally abnormal. So maybe it's a problem of a developmental problem. So the cells that make up his brain originally, there might be changes in the number of cells or the way they migrate or the connections they make. So, and we got some insight about this when scientists actually made mouse mutants, which are genetic models of trying to mimic some of these human mutations in ARX. So shown on this slide, we figured out from many, many studies, including collaborators of ours, Jeff Golden, who's the chair of pathology at Harvard Medical Center, they figured out that ARX is one of these genes that are in this transcriptional network. So it's downstream of master regulators that make these inhibitory neural cell types, and it controls other genes as well, so both in a positive and a negative fashion. So it binds DNA, but these polyalanine tracks, these repeated domains, maybe disrupt the DNA binding itself. And then this particular gene is located in a part of the brain, as shown here, that is where the source of the stem and progenitor cells that give rise to these neurons that end up migrating into the cortex. Okay, so from these mouse mutants, scientists have figured out that, here on this slide, we can see actually the pattern of this protein. In the wild-type mice, they look normal, they're nuclear, and in the right, these right inhibitory neurons. But in the mutant mice that have these repeated alanines, there's fewer numbers of them as shown here, and the distribution of the protein ends up in the wrong place. So it's like cytoplasm as opposed to the nucleus, where it works. So we've gained insight from a lot of these ARX mouse mutants. What I'm not showing, I don't have time to say, but the animals themselves have seizures and they have memory deficits consistent with patients with this type of genetic epilepsy. So it's very exciting. But one of the things with a limitation we've heard already this morning is there's differences in mouse and human brain, potentially, both in the way it forms and the way it functions. So that's something to consider. possibly why we still don't really have good medicines to treat patients like Hsieh. The other question is whether these mouse models, they're not very easy to do drug screening because it's like you have to do a lot of breeding and it's hard to sort of find the right medicines that tailor-made for the patients. So we were kind of excited to use some of these stem cell approaches to try to get at maybe what these differences are and maybe begin to use that for screening drugs. Okay. So I don't want to belabor this point because we've already heard some of the slides this morning about how there's differences in mouse and human brain development. So besides the enormous size difference, but there's also this complexity differences. And on this cartoon, I'm just kind of showing you, one of the cell areas that might be different between human and mouse based on studies is, in addition, these stem cells originate in the ventricle and then they differentiate to these immature and mature neurons that migrate into the cortex. But in humans, there might be new or different populations of stem and progenitor cells contributing to the diversity of the cell types in the cortex. The other potential difference is the way the cells, the stem and the progenitor cells, they're born and then they move or migrate into the cortex. So in the mouse, it's generally thought that they're born in these areas, the ganglionic eminences. It's really the bottom part of the brain, and then they migrate to the top or the dorsal part. But in human, in addition to that type of migration, there might be a different type of migration, sort of more locally in the cortex itself. And so from these are just another type of the possible differences between rodent and human brain in terms of how they develop and also the way they migrate. Okay, so keeping that in mind, so our idea is as follows. It's pretty simple. So we take our patient, for example, Hsieh, with known set of genetics based on its whole exome sequencing or even whole genome sequencing. So the gene of interest or the mutations identified, we can that'll become relevant later when we think about the precise control for Hsieh. But in the meantime, we're also recruiting patients that are healthy, that are matched for age. And in this case, since ARX is a gene that's on the X chromosome, only boys are affected, and so all of our healthy controls are also boys of the similar age. So we use this basically, we put a series of transcription factors into the blood sample. So we take blood, so it's very simple, or we could take a skin biopsy, very easy to obtain cells, and then we can basically put 4 transcription factors that was described by Yamanaka and colleagues that first identified 2 reprogram terminally differentiated cells back to a pluripotent stem cell, which is a cell that could become any cell type in the body. So now we have cultures of these pluripotent stem cells in a dish. We can expand them and make sure they're of normal karyotype, and then ultimately for the idea of generating these organoids, which I'll tell you in a second. But the idea is these are fully scalable, these 3-dimensional structures that resemble these brain-like tissue. They're very small. They're only about 3 or 4 millimeters. So you can imagine putting a single organoid per well of a multiplex plate for the eventual purposes of toxicity testing or drug screening towards personalized medicine, which is hopefully our ultimate goal. Okay. So here, the next 2 slides, I'm just going to show you some of the types of analysis that my lab and others do to analyze these organoids. So they're very, very small, and they're 3-dimensional. So I'm just showing you 2-dimensional sections to represent some of the data that we get. So for example, we actually adapted protocols from Dr. Sergio Pasca from Stanford University. But these stem cells, they self-organize, and if you add factors that recapitulate the way the brain tissue normally develops, these sort of growth factor signaling pathways, you can bias the stem cells into neural fates that will ultimately give rise to the neurons, oligodendrocytes, and astrocytes that comprise the central nervous system. And you can also give additional factors, so it kind of knows to be the dorsal or the top part of the brain, these cortical cell types, or the ventral, inhibitory neurons. And so the details are not that important, but that's what some of these this is what this slide is showing. So we take these stem cells, we plate them, they form these spheres, and then use, over a period of time, up to, so far, we've done some analysis to about 6 months, but we still have organoids going for as long as 9 months or 12 months. And so then we're also doing studies like protein analysis to define the cell types, both in a biased way where we could pick certain neuronal cell types to look for, but we also want to do this in an unbiased way using transcriptome analysis. Okay, so here are just some cross sections through these types of organoids, showing you the different types of brain cell markers in a control patient. And then we also can look at, other markers of this, the inhibitory neurons shown here. So they express, inhibitory neurotransmitters and also some interneuron neurotransmitters. Okay, so then going back to our patients, currently we've recruited Hsieh and also other patients and controls. We always verify that the stem cell lines are normal using a variety of different assays to make sure they're pluripotent. And so now what we're doing is trying to see if we could really study epilepsy in a dish. And so epilepsy is, if you break it down, we hypothesize that maybe there's going to be this excess activity will come from several different features or aspects. For example, maybe, as I already alluded to, a change in the progenitor cells, maybe fewer of these inhibitory cells or more of these excitatory cells, the movement or how the cells form and find their ultimate targets, their morphology, and also their function at an electrophysiological level. So we've started to kind of systematically go through these. And so, for example, we can also see where is ARX expressed in the cross sections from the brain organoids. And so far we found that in the patients, in the control, there's a lot of this, the protein, and it looks like it's in the right place. But the organoid section from our patients appear to have reduction of this protein. So, and that's something we're still characterizing and trying to quantify. So that's consistent with, what is observed also in the patient samples. Also, we can look at, the number of the cells and the migration of the cells. So this slide just generally shows you, one of the protein markers we look at, NKX2.1. It's a marker of these inhibitory cells. There appears to be fewer numbers of those in our patient-derived organoids. We can also use unbiased methods. So we heard from this morning, we could just take this genome-wide approach based on the genes that they express, take the individual small organoids, dissociate them similarly, and measure gene expression as an approximation of what the cell types are and what genes are dysregulated. I don't have anything to report except that we're just starting to do this. Okay. And then we could look at the movement of the cells, and one way to do this is to label the inhibitory cells with a GFP reporter, and then you can fuse the top and the bottom spheroid together and actually watch this movement. And then finally, you can look at the activity of these organoids themselves. And one way to do this is actually to put these organoids themselves in these wells where there's electrodes that are sampling the neural activity. And you can do this in real time. They're called multi-electrode arrays. So the organoids, after about 4 months, start to fire and have action potentials reminiscent of neural activity or a network activity. And so here I'm just showing you, they burst and they spike. And then, so what we found is that organoids, at 4 months compared to 6 months, there seems to be more activity over time. And we're starting to also compare the activity of organoids from our patient versus our control. And at this point, it looks like there's some differences, and we don't really understand, what they mean, but we're still analyzing that. Okay, and then ultimately, so what I told you today was just comparing between healthy and control patients, but because we have the gene in mind, specifically the mutation, we could use gene editing using CRISPR to correct the patient mutation, so we really have precise isogenic controls. Okay, so finally, I think since I'm running out of time, I just want to wrap up and say that, so these are just sort of tentative conclusions using these brain organoid models derived from patients and controls. And we think we're starting to at least have these in vitro-based models to access human development and potentially disease in a way that would be difficult and costly and limited otherwise. And ultimately, we really want to try to improve treatment options. So these are just a small table of some of the medicines that are used to treat kids like Hsieh. As I mentioned, he failed 4 or more medicines. It's also quite empiric how they determine the doses and of which compounds to use. So we're hoping that in addition to the possibility of doing drug screening in a sort of a high throughput manner, that we could also just go back and sort of in a less empiric manner use these IPS-based model to helpfully guide precision or more precise dosing and medicines ultimately, for personalized medicine. Okay, so really the take home is we think that using these stem cell-based approaches, it may represent exciting, approaches that complement existing models. They also can lead to insight and biological discovery for, maybe human cells and how they work and function, and then for therapeutic discovery. And finally, just want to thank the people that did the work. And also really, this is really a team. It's a team with basic scientists, neuroscientists, stem cell biologists, and clinician scientists, and including people from UT Southwestern, UT San Antonio, and Harvard and Children's Hospital of Philadelphia. And I also want to just, one last thing to say is that we want to really create a one-stop shop for this type of technology. So we've established a core facility at UTSA that basically starts with the patient, and we have an IRB approved to create these stem cell lines, and we can even help you with differentiation protocols, such as the organoids we talked about. I'm also working with a team at UTSA and UT Health San Antonio that does medicinal chemistry and drug screening to sort of apply this to this technology. So thank you very much.
We have time for a quick question or two.
Yeah, a quick question about the membrane or the overall, the morphological properties of these neurons that hyperexcitable. Like, do you suspect any changes in the mechanotransduction properties like cell elasticity or the stiffness of these membranes that actually stick together all these ion channels that you suspect in these genetic epilepsy?
So I think the question you're asking is, what is the mechanical composition and structural properties of the neurons within these organoids and potential, and I think to extend that, maybe how similar or different are they to other types of neurons found in the brain? I think those are really important questions. And one of the first things we're trying to do is actually do single cell patch clamp analysis on the organoid-derived neurons directly in slices from organoids. And at least my postdoc in the lab who's doing this work says they're very, very different. And we are still trying to figure out what's the composition of the different ion channels.
Jenny, a lot of brain malformations lead to epilepsy. And I was sort of, in a way, it complicates the analysis of what actually is a seizure, because the anatomy is so messed up. And I was wondering, among those many genes that are genetic causes of epilepsy, how many have essentially otherwise normal development and cognition and so forth? It seems like that might be a really fruitful way to actually get at mechanisms of epilepsy that could be druggable.
Right, absolutely. So I think the debate, so I think what Mark is saying is how, maybe using this approach, what you're going to find is different in every case. So on that list that I flew through, many of the mutations are ion channels, obviously. Like, for example, sodium channel, which is very, very common in Dravet syndrome. And so that's also another childhood epilepsy. So in those cases, you're right. There's less of this developmental property. So we sort of strategically made a choice to go after some of these developmental patterning types of deficits. So I'm not really sure how they compare.